Hello, everyone. So we're about to start with day three of session three, the first workshop for today's afternoon. Sorry, we're starting a little bit late. We're just one of us is sort of trying to address some AV issues. Yeah, so I hope everyone had a nice lunch. And yeah, I think we'll just get started. I'm gonna quickly share my screen just to give a sense of the agenda here. So this is the agenda. Okay, so hopefully you can see my screen. So this is where we are, workshop one or workshop three, intro to transcriptomic data types, including single cell and bulk RNA-seq. This workshop will be led by a postdoc in my lab, Dr. Sonny Chen. And then the next workshop will be using the tools that we made use of in the first tutorial to understand cellular changes in major depression using bulk and single cell RNA-seq. And so that will be led by Kian Arbabi, a PhD student in my lab. So I'm going to stop sharing, and I will allow Sonny and Kian to introduce themselves. So let me see if I can close and stop sharing. Okay, great. So we're all here. Just one sort of point of complication is not quite sure if Sonny can you still hear us? Okay, so Sonny can't hear us, so I'm going to be communicating with him via Slack or some other internal messaging system. But I think the tutorial will work okay. So Sonny, I'm going to ask Sonny to introduce himself.
Okay, can people hear me? Okay, great. So hopefully Shrija has explained that I am basically deaf right now. I do not hear any audio, but we will do what we can to go forward because the show must go on. Now, is it possible to maybe get a poll going to see how many people are following on Binder versus Docker, or if they're going to be using nothing at all, or if they're just following along?
So
And for those who are just following along, who do not, who maybe don't, for any number of reasons, might not want to do any of the coding or following along, I will post the link for our GitHub repo. And then specifically, you can see at the bottom in the README, there is a clickable link for tutorial 1, single cell transcriptomics. And while I'm at it, I can probably just give the link for that HTML file as well. So when you click on that second file, you will, like I implied, go into a published document of our RStudio, of our Markdown file. So just taking a look at the whole, just following along are the majority, that is great. There are 3 people using Docker and 4 people using Binder. Okay, that's great. So Docker people and the people that are just following along, you will be fine. For people who are using Binder, if you have experience with R Markdown, what you can do is basically for that Markdown file, you can just tell it to start running all of the code. And it'll just go through all of the chunks of code and kind of just give you all of the output. Everything will be saved into your environment. Do not do that, right? I would generally not recommend doing that for a tutorial anyways, but especially for the people using Binder right now, do not do that, because there will be a function that either that takes a lot of memory and you'll either hang or perhaps your Binder session might crash. That being said, hello everyone, welcome to the tutorial. This is very jarring to speak into the void, but we'll go through this. The TAs that are helping with this, I believe, are Michaela and Keon. So maybe if the people who are following along on Docker or Finder can probably turn to them for help, either on Slack, in the chat, or in GatherTown. Just in case the Binder folks, and maybe the Docker folks as well, are still loading as well, I'm just going to do like a really quick recap about some of about what we're going to be doing. Shidre has basically gone over what we're going to be doing, but since we've had lunch and maybe we're in that post consumption kind of drowsiness. So just to recap things, ultimately what we're going to be doing is comparing cell type portions between people with MDD and people without. The samples, to do this, we will need bulk RNA-seq samples and single-cell RNA-seq sample data. The bulk RNA-seq, so RNA-seq data that comes from essentially chunks of tissue that we get from participants. That is essentially the RNA-seq data that we will basically use to estimate the different proportions of cell types in each chunk of tissue. And that will mainly be the second tutorial. To do that kind of estimation, though, we need to actually define the cell types that we'll be looking at. And we will also need the genes that best defines those cell types or best represent those cell types that we call markers. And that is basically what this tutorial is about. Starting basically from essentially raw RNA-seq data. Two general points to point out at this stage. One is that we are showing a variety of tools and code, and it is very likely that when you look through, for your own teaching and understanding, when you look at other resources, some people might say some other tools, some people might say do things slightly differently here or there with slight tweaks, and that is all very normal. We are not, no, it is not that we are wrong or they are right or vice versa. It is just there's a lot of nuance in these steps, such as what tools people use. And it is, again, just very normal for people to use different tools. I will explain as we go along of why we do some of the things we do. But overall, the general principles are the same. The second general thing I want to mention at this point is that we are going to be touching upon a lot of techniques that are related to more advanced math, more advanced algebra. And depending on your background, depending on your training with different levels of statistics or linear algebra and so on, you might be very familiar with these things, or you might be very unfamiliar with these topics, and that is fine. I will be going through these things at a fairly superficial level. If you are not familiar with some of the techniques that we'll be talking about, that is completely fine. If you are confused about any particular concept, feel free to ask a question. I will try to go slowly and be able to pick out if anybody has any questions about those things. But generally, just do not be intimidated. Do not feel bad for being confused when there's any particularly higher level mathematical concept that we're touching. We're only going to be talking about those things in a very general level, and I will do my best to be very clear about whether something is maybe a bit more advanced, and we're really only going to stick to a more general level of detail. And for things that are a bit more complex, for people who are familiar with those things, I will try to say a few comments for those who are a bit more familiar with these techniques as well. So hopefully that is enough time for people to load their code for those who are following along. So I will now try to share my screen. Interesting. Okay. Can people see my screen? Perfect. Thank you. Yes, very good point, Kian. I will zoom in. Okay. Are people good with this size of font? Could be bigger. Okay. Is this font size good? I'm going to continue since nobody's saying no. It's going to be good. Okay. So this is an R markdown. Oh, and again, for we've seen this. We've seen R markdowns with both Sejal and Marta now. One thing, of course, that is very important, perhaps objectively the most important thing, is if you go to Tools, General Options, you can switch to some kind of night mode, dark mode, right? Because that is, of course, the most important thing. Jokes aside, I like using some of these other appearance themes to really help distinguish between the different text and the different labels and code and the commented out code. So we've seen R markdowns multiple times now. So that is really what this first section is about. Again, just in general, a markdown document is where we have basically notes and text and paragraphs combined with chunks of code. And not only that, oftentimes I will see as we go through this, when we run these chunks of code, there will also be the output of these code that comes up at the bottom of these chunks of code. For this, so I gave a bit of a general overview about what we're doing with our two tutorials this afternoon. For this particular first tutorial, we will be going through some of the very basics of RNAseq data. We'll talk about reads and counts in a counts matrix. We're going to import and work with this data, do normalization, perform dimensionality reduction. And that's, again, one of those perhaps more advanced concepts that we'll talk a bit more about at the time. But ultimately, the goal for this is to simulate the finding of different cell types and also finding the genes that best describes or best differentiates those cell types. So we are going to start by loading packages. We are going to load tidyverse. Again, this is to help just a collection of tools to really just make it easier for us to work with our data. The main workhorse, if you will, of this tutorial, the main workhorse that we like to use in our lab to deal with RNA-seq data, in particular single-cell RNA-seq data, is Seurat. This is a very common package, and it's very commonly used. It's well supported. The thing I like about it, or we like about it the most, well, I like about it the most, and the thing that I'm sure a lot of people really appreciate about it, is that it is very well documented. There are a lot of good, very good tutorials and vignettes for different aspects of all the different things you can do with Seurat. MAST is a statistical package. It is the main statistical test we'll be using to look at differential gene expression, which is the method that we will be using to get our markers. And then I'll speak a bit more about it when we get to using it. And finally, NITR or knitr to basically help with some of the markdown parts of this document. The data set we're working with is a downsampled data set from the Allen Institute. We are looking at human cells from multiple cortical areas. And it is freely available from the Allen Institute. Of course, to make it so that everybody can hopefully run this code on their computers, we downsampled the number of cells and the number of genes to use for our demonstration. So first, we will take a look at the metadata of our data set. So this format is essentially every row is a cell, and we have a lot of columns with different variables, different attributes of each cell. So for example, this first cell is part of this cluster, part of this class. It is a glutamatergic cell. And we can see the donor, the sex of the donor of the tissue that ultimately gave us this data. As you can kind of see from the fact that there's kind of like cell identity labels here, this data from the Allen Institute already comes pre-clustered, right? So the Allen Institute has already done a lot of the legwork to identify and to find and identify and annotate all of the different cell types that we will be working with. We are still going to be going through something similar, a similar process for as a simulation, as a demonstration. But again, this is just a note that we ultimately will be working with these types that are defined by the Allen Institute. And indeed, we also have just a lot of other information about each cell and where it comes from and its characteristics. We are going to set the row names for the metadata. And so I'm going to run line 72, but also 74 to show that table. Okay, so this is set to row names as false, but essentially we do want to also set these sample names as the row names. As I'm sure for any of you who have used any kind of bioinformatics tools or statistics tools or math tools, like you know that for any particular package, for any particular custom function, like you need to know the specific format of the data that you need for getting that tool to work with the data. In the case of Seurat, and in the case of a lot of packages working with RNA-seq data, they need some way to connect the sample name and their metadata to the sample name in the actual counts, right? Your actual measurements of RNA. In Seurat, we need to set the sample names to its row names. Seurat is going to look for the row names of our data, and that is where it will know to look for identifiers to match with our actual count matrix. It might be different for different packages, but this is, of course, something always to be aware of when you're working with any kind of package or tool. So speaking of matching with our count matrix, we can load that in as well. So this might take a bit longer. The general concepts and the general concepts and the contents have kind of been demonstrated and mentioned by Sridhar earlier. this is our count matrix. We're at the beginning of our count matrix. Every sample, in our case, which is a cell, individual cell, every cell or every sample is a column, and every row is a gene. In this count matrix, the numbers in these cells are essentially quantified measures of the number of RNA fragments or reads that have been aligned and detected for a given gene in a given sample. And again, in our case, a sample is a cell, but the structure of bulk RNA-seq is pretty much the same. You can still have your columns as samples, but in the case of bulk RNA-seq, every column or every sample would be a chunk of biological tissue from a subject, or like if you're doing an animal study, it would be from a particular animal, for example. This number of 50, so this number of 50 means that in this cell, within this ID, there are 50 RNA fragments that were measured, that were aligned and assigned to this gene. So the A2ML1 gene was detected at a level of 50 fragments in this cell. It was detected, there was only one fragment of this gene that was detected in this cell, and then there were 71 fragments of this AAAS gene that was detected in this cell. Okay. So that is our data. That is our actual measurements of genes in our samples. So now we're going to go down. So now we're going to be actually using Seurat. And it is very simple. We're just going to call the function of create Seurat object, and we're going to give it our counts matrix, and we're going to give it our metadata. And again, if this function, the overall Seurat package, is able to associate the metadata associated with every cell to the gene expression measurements for every cell through the row names, or through the row names of the metadata and the column names of the count matrix. If we take a look at our Seurat object, so we can just run the Seurat object code, you can see that there are 31,563 features across 893 samples. So you'll notice that these two numbers match the actual dimensions of the data that we imported. And again, it's just showing that things are working and we have our data now in our Seurat object. One thing to point out, features is what Seurat essentially calls genes. This probably has to do with how, if you think about what the definition of a gene, it's a region of DNA, but it has a functional purpose. But what happens if there's a region of the chromosome, there's a region of the DNA that we detect RNA for, but we don't know if it codes a protein, we don't know if it's a microRNA, we don't know what it does. So at that point, if we don't really have a concrete function, is it a gene? Is it, at the very least, we can probably agree that it is not like the traditional definition of a gene as defined in the context of the central dogma. So because of that, again, we kind of use the word features for when we were looking at like genetic, when we're talking about regions of the chromosomes and the regions of the genome that we have detected, that we have divided into areas and that we have detected RNA-seq fragments for. But for simplicity's sake, you can think of features as genes. these two lines of code, again, just kind of shows us where the metadata that we imported and the count matrix that we imported is stored in our Seurat object. That is Su_obj here. So we can see this for the metadata, and we can see this for the RNA counts. Overall, this is probably a good point to say that a Seurat object is essentially a list that has matrices, that has vectors, that has a whole bunch of things that are related to our project, that are related to our analysis, that are related to our data. All of this, again, is basically stored in our Seurat object with different regions, different slots for different types of data and different formats of data that we can add to our object. And we will get to that a little bit more when we talk about dimensionality reduction later. So one of the first things we do with single-cell RNA-seq data, and really with all RNA-seq data, is normalize our gene expression, normalize the measurements in our data. Now, there are a few different ways for this, but in general, you can think about the situation where you have two cells, and for any number of reasons, maybe technical, one cell just has more RNA captured compared to the other one. So if it just has more RNA captured, it might have more gene A transcripts captured compared to the other cell. But this increase in gene expression would not, this difference in gene A expression might not necessarily be anything biological. They might be from like the same kind of cell. But because of, again, due to any number of reasons, we just detect a lot, we were just able to capture and recover a lot more RNA from one cell. Statistically, there might be a difference between the two, right? So this is why we normalize our data. We are going to essentially take, there's other things going on here, but essentially the main point is to take the measurement of each gene and normalize it to all the RNA-seq, all the RNA measured for a given cell. So that is what we are doing here. Now, one thing that, of course, for people who are used to coding, even at a very basic level, right? One of the things we're always told is to, right, be careful about overwriting your object. Be careful about overwriting, your data. You don't want to, you want to be very careful about assigning, in this case, putting in a Seurat object and seemingly overwriting our object. And what I wanted to say here is, obviously, if anybody's careful about what they're doing, that's not an issue. But I do also want to make the point that a lot of these Seurat functions, or Seurat objects, a lot of these functions basically have outputs that get stored in one of those other slots in the Seurat object. So notice how there's graphs, neighbors, reductions, and all of these are empty. Well, oftentimes when we use a Seurat function with a Seurat object, and we assign it, send it back to the Seurat object, like we are technically overwriting it, but we're almost always never really overwriting anything that's already there. Most likely, obviously, unless you're running the same function again, right? Then it definitely overwrites the previous output of the function. But generally, it would save it in a different section of your Seurat object. So in this case, what is happening is, notice how we had assays, RNA counts. So this would be our raw data that we put in with integers. But now we have assays RNA data, right? So our counts, like the counts section of this start object has not been touched. But this normalized data function has now stored all of those normalized data in this other part of our start object. Okay, so you can see we no longer have integers, we have our normalized data. And this gets us our counts per million. Now we are going to do dimensionality reduction and clustering. This is a simulation of what somebody might do to try to look for groups or clusters or subclasses or classes of cells from RNA single cell RNA-seq data that they just obtained. Again, the Allen Institute has already done a lot of this work with their own methods and their own kind of tweaks. What we are going to be doing is we're going to be going over a set of steps that basically does that for demonstration purposes. So to do this, for the first thing we're going to do is dimensionality reduction. And again, this is that, again, more advanced kind of math topic that we are approaching here. In general, well, you can think of what we're going to do is summarizing all of the information in our RNA-seq data, where we have for 893 cells, information, measurements, numbers for 31, almost 32,000 genes or features. We are going to use dimension reduction to basically summarize this data to keep as much, to represent this data of 32,000 genes. We're going to try to represent it as faithfully as possible to maintain the structure, to maintain the variance, to maintain as many characteristics as possible of these 32,000 genes into a reduced number of variables, a reduced number of dimensions. As I'll show in a bit, we're basically going to have 50, like just by default, it kind of gives you 50 of these reduced dimensions. So you can think about it as summarizing 32,000 genes into 50 variables. And we're going to do this specifically with a principal component analysis. So those 50 variables are called principal components. To do PCA, first thing we need to do is scale data. We need to scale our data because you might imagine certain genes just has a lot more expression and a lot more variable expression than others. So then in which case, well, if one gene has a higher variance, it will be treated differently than another gene with very low variance. So if a gene has, let's say, a range of expression of like 0 to 10 versus a gene that has a range of expression from, let's say, 0 to 1,000, the 0 to 10 will be weighted differently compared to the 0 to 1,000. And we do not want that. We want the PCA to treat every gene equally. Now, again, for those who are more advanced, for those who have like a very specific question, a very nuanced case, they might make the argument of perhaps not treating every gene equally. But for the most part, very commonly, and for us for sure, we are going to scale our data so that they all have equal variance and they're centered around 0, which will help the PCA process. This is the actual code that will run the PCA. Do not run it yet, especially the binder people, do not run it yet. But before we do that, we're going to find the most variable features. we are going to find the genes that is the most variable in expression among our cells. This is an optional step, and it's a common step, but we are mainly doing this. The main reason we are doing this is that when we actually run our PCA, we're not actually going to summarize 32,000 into 50, mainly because of computational reasons. Your memory, the memory in your binder or Docker, or even if you're following along on your local laptop, could be very limited. So and actually, if we did this without doing the find variable features, the binder instance will definitely hang and maybe crash, or it might just take a very long time, but complete if you're lucky. So we're going to run this line of code 151 to find the genes that are the most variable. So again, this is going to be a bit of a shorthand. We're going to use these 2,000 genes to represent the 32,000 genes in our original data set. And we're going to run a PCA based on those 2,000 genes. So we can run that now. This might take a little bit of time on the Docker or binder. But ultimately, what we have are these reduced dimensions from, in our case, the 2,000 most variable genes. So here we can kind of see where our piece, we can take a look at our PCA results. And again, notice we ran, so I believe this is a strat specific function. I believe the actual PCA from base R is different. So the when we run PCA with a strat function to strat object, again, it surprise I mentioned before, we are now storing the output into another part of this strat object. So let me just refresh that, and you'll see now reductions where it was empty before. Now we actually have some reductions at PCA, and particularly we have our PCA results. So our PCA results, we can basically see the relationship of every cell. So we can have an actual PCA value for all of our cells in these cell embeddings. And we also have what are called feature loadings, and you can think of them as correlations between every gene, every one of those 2,000 genes that we input to the 50 principal components that we output. What we are showing with this line of code, again, are just like the, for each principal component, again, those reduced variables. The positive genes, so the genes that are most positively correlated with the first, with each component, and the genes that are the most negatively correlated with each component. And we can visualize all of this information with a heat map, which is what this line of code does. We are only going to show for 2 dimensions, so the first 2 principal components. And this heat map is only showing 300 cells out of our 800, and 50 features, again, or genes of our that went into this. So we can see in general, in this kind of situation, what you have is yellow for high expression and purple for low expression, or yellow is high. The purple for negative. And you can basically see how for different cells, different groups of cells, you have these genes that are well represented by given component. So that is about where we will go for principal component analysis. And Keon will be going through principal component analysis as part of our cell type deconvolution or our method for estimating cell types from bulk RNA-seq data in the next tutorial as well. But we are going to end here with this kind of illustration of just basically seeing all of these numbers that illustrate how different the relationship of different genes and different cells in the x-axis, genes on the y-axis, and their relationship to the different principal components. So you can see that, for example, this principal component might really represent these genes well because of their highly correlated or anti-correlated nature. Now we are going to find cell types. We're going to find and define cell types and groups. And all it really is cluster, right? We're going to find, if you've worked with cluster analyses or finding clusters in a different kind of context, we're going to basically be doing something very similar. We are not going to be using, for example, a centroid-based clustering method. We are not, but somebody could be using something like that for clustering. For those of you who are not familiar with clustering, I will go into a little bit about what we are doing. But again, it's just for our purposes for this tutorial, it is sufficient to just kind of understand things at a high level, at a high general level. So in general, what we're going to be doing is this will probably be easier with an illustration. So I'm going to run these lines of code. We're going to visualize it, and then I can explain a little bit about what we did. So first, we're going to run this find neighbors function. And then we're going to run the find clusters function. After these two steps, we've already basically, Sirac has already given us clusters or groups of cells that are the most similar that it has detected. We are going to visualize everything on a UMAP plot. You can think of UMAP as another dimension reduction, but now you can think of it as we're going to take our 50 variables from PCA, and we're going to reduce it into 2 variables that we're going to plot on the x and y axis. And that is what this plot is. Right? So here we have, again, we have our 2,000 genes summarized into 50 variables. And these 50 variables, you can think of it as being summarized into 2 variables for plotting purposes, for visualization purposes. And we're plotting these variables on the x and the y axes. So where we had PCA1 and PCA2 before, now we have UMAP1 and UMAP2. We're plotting these values for every cell. So every dot in this plot is 1 cell. So for every given cell, its measurements of 50,000 genes, we selected 2,000 genes from that. From those 2,000 genes, we reduced to 50 variables that we use to find clusters and groups. And from those 50 variables, we are reducing again to a 2-dimensional space to really just for a visualization, really for illustrative purposes, for a demonstration. So we can see these our dots. Again, every dot now has essentially, you can think of it as they have all of the gene expressions summarized into 2 dimensions. So dots that are closer to each other then would have very similar gene expression profiles, right? Cells of the same group might always have high gene A, low gene B, and 0 for gene C, and so on and so forth. These numbers are the labels that Sirac has detected for cell types and cell groups. And a big part of people who do this kind of work, a big part of the work at this point, for people who are doing this de novo, for people who are doing this from scratch, is then they need to kind of dig into each of these groups and say, oh, are these neurons? Are these non-neurons? What's happening here? Right? We don't need to do that because we'll be working with the Allen Institute definitions, but that is where people typically go from there. Now, so now that we have this visualization, I want to maybe explain this find neighbors and find clusters a bit. Find neighbors and find clusters works on PCAs, works on those 50 variables, not these 2 variables. But we can just, again, for illustrative purposes, for explanation purposes, you can just imagine that it's PC1 and PC2 rather than UMAP1 and UMAP2. What find neighbors does is essentially find all the cells, so every dot in this 2D space or multi-dimensional space for those principal components. But for every cell you can that have similar gene expression based on the 50 dimensionally reduced variables, draw a line between them. So in this case, let's say I draw a line between this dot here and another dot here, right? Because they're the closest to each other. And we do this for every cell, and we draw this for a certain number of nearest neighbors, right? Or so every cell is going to have a certain number of lines connecting it to that same number of other cells that has a similar gene expression profile based on those 50 variables. So that is basically, you're just establishing that relationship, right? You're just establishing which cells are close to which cells. And maybe you can do a weight, right? So the closer the 2 cells are, they might have a higher value. The further the 2 cells are, but still, close, relative close, right? They might have a lower value. You can do a weight for this relationship. What you then do is use this established relationship data, and you put into find clusters. And what you can think of this as doing is basically maximizing the number of lines between cells within a group and minimizing the number of lines between cells outside of 2 groups. So you can imagine all of these cells having a lot of lines, imaginary lines, kind of just connected to each other, right? And if we define this as a group, well, we're going to have a lot of lines between cells here, and we're going to have very few lines between, let's say, this pink cell and this yellow cell. That is what the clustering part of the function does, right? So for every cell, this neighbor function, for every cell, we're going to draw lines to the closest, to its most similar cells. Those are the closest neighbors to that cell. But the find clusters function, we're going to group cells such that all of the closest neighbors are together. All of those lines that connect neighbors, they're going to be within a group, and as few lines as possible between cells from different groups are going to be, we're going to minimize those connections of cells between groups. And again, that was done at the PCA level, so we're going to run a UMAP to basically summarize everything into 2 dimensions, and that's what we see here. So hopefully that was fairly clear for people who have some general idea about clustering or not. For those who have perhaps less of a background in math, just know that basically we summarize all of those 32,000 genes into 50 variables or 2 variables, and then we just want to find all of the dots that are closer together. And we do this in a mathematically robust and rigorous way. So as I mentioned, most people at this point, they would then maybe need to do a lot of digging to see, cell group 5, what are they? Like, are they excitatory cells, inhibitory cells? Are they not neurons, right? People would probably be doing something like that if they were doing this de novo for themselves. Again, for us, we are lucky to have the definitions that are already defined in by the Allen Institute in our metadata. So we can just take a look at that. In Seurat, when we want to do something between groups of cells, Seurat uses an identity. or an active identity. The way to specify this is basically use this ident function. We're going to put strat in our function, and then we just give it the column name, the column of the metadata. So in this case, we want the subclass labels from the Allen Institute to be the active identity that we use for our strat object. This means that when we tell strat to give us group means, when we tell the strat to do a plot, when we tell strat to give us different differential gene expression between groups, we're going to do it for different subclasses. If I put class here, it will do it for different classes. If I put subclasses here, it will so classes will be like excitatory versus inhibitory cells. If I do clusters here, it will be between clusters. If I put donor sex here, it will group the cells by male donor versus female donor. So again, all this is, and this is very commonly used in Sriracha, right? Because this is kind of like the key thing to define the groups that you're looking at. So now we have these, I think I ran a choice, that's fine. Now we can take a look at the same plot that we just looked at, but with the Allen Institute labels. So first of all, we see some very good clusters, right? We have parasites that are basically by themselves, endothelial cells, astrocytes, microglia. We have some groups that are a bit closer together. SST cells and PVOP cells are inhibitory neurons. So them being close to each other, but still fairly well defined as groups, that looks pretty good. These are excitatory neurons. So they are also very close to each other, but now we kind of see these cells, these groups being perhaps a bit less well defined. So you see, for example, blue cells mixed with these orange cells. And we see this with these inhibitory neurons as well. Why do we see this? Well, again, we didn't do, first of all, we're working with downsample data, right? So we didn't have all of the data that the Allen Institute would have used to their groups, to do their dimension reduction, to do their clustering. We also didn't use all of their genes, because again, we downsampled the number of genes. And we also use a different method than what the Allen Institute uses. Now you can, yes, so technically they might have used different like mathematical methods for doing it, and that's true too. But there are also other methodological differences that different people use for clustering their diagrams, for clustering their data. So one common example would be, for example, and I know the Allen Institute has done this for some of their data sets, for at least some of their data sets. What they did was they took out sex-specific genes, for example, from their clustering method. And when they do this, because you can imagine, if you include all of those sex-specific genes, right, the genes that are high in males or high in females or low in either sex, what you might have is, for example, male parasites here and then female parasites here. Parasites. So yeah, there's a lot of considerations, a lot of nuance that thankfully we don't need to go into, but all of this is just basically to say there are a lot of nuances here that could explain why our diagram is a bit different from the groups that the Allen Institute have. But for the most part, we can also be fairly reassured that most of these cells are grouped pretty well and pretty independent. So that is a simulation of what somebody might do to find groups of cells, to find clusters of cells. Now we're going to find cell markers. And again, we are essentially going to look for genes that are significantly different, or gene expression, or genes whose expression levels are significantly different in one group versus all of the other groups. So again, we're working with subclasses. This is just in case. This is just to be safe. We want to look at subclasses. And we can take a look at all of the subclasses that are available in our data set. So in Sridhar's talk, you mentioned telencephalate, acetinones, inhibitory, and we have our endothelial astrocyte non-glial cells, for example. This is the line of code, right? Warning, warning, blaring, signaling here. Try to be very clear here. This is the line of code that will cause people who are using binder to either take a long time or hang or crash. Do not run this line of code or this bit of code if you are using binder. I am not using binder, so I'm going to just run it. And you can see even for me, it takes a little while. What this is doing is essentially for every gene, it's going to compare its mean expression in a given cell type, in our case, given subclass, versus all the other subclasses grouped together. So we're gonna compare gene A's expression in layer 5, 6, and P cells versus all of these cells. We're going to look at gene A's expression in IT cells versus all of these cells and these cells. Right, so all of the other cells, all of the non-IT cells. And we're going to do it for this cell versus all the other cells and so on and so forth. It is systematically going through every subclass. The specific test we are using is MAST. This is a specific statistical text that takes into account the fact that we are using single cell RNA-seq data, so it might have a lot fewer genes that are captured because, again, just the amount of RNA material you capture from a single cell is less than you might capture from, that's for sure, from a chunk of tissue. So MAST essentially takes into account the number of genes detected in every cell and works that into the statistics of our differential gene expression testing. Some of these other thresholds are also, we also use to basically use a threshold to look to determine which genes we are looking at. You can imagine if a gene is not, so if a gene is high in IT, but it's only high by 10% compared to all the other cells, well, we might not be so interested in looking at that cell. If a gene is high in IT, and maybe it's like 10 times higher than when you combine all the other groups together, or maybe 100 times higher, but you only detect that gene in maybe like 2% of IT cells, right? Maybe we don't want to look at that gene then, right? Maybe there's something weird going on in that situation. These thresholds are basically where we can use numerical values to actually put a threshold cutoff for just how highly enriched a gene needs to be for us to do that stats test, and how many cells within a group the gene is detected in for us to do that stats test. We are using fairly high thresholds here, and part of the reason for that is to basically, again, for memory issues. We can take a look at the outputs of our final markers function. And we can see again, for each gene, it has a p-value, a statistical significance value, the chance, as Dan explained it, a summative number that that this gene is highly enriched in one cell versus the other cells, in one cell subclass versus the other cells, and not due to chance. Or rather, due to chance, So if it's a low number, then it's not due to chance. So this is our fold change. So just basically how enriched the expression is for this gene in this subclass versus all the other cells. What percentage, or fraction, what fraction of this of the given subclass expresses this gene? So in this case, 100% of layer 5, 6, and P cells express this gene at a level greater than 0. In contrast, 41% of all of the other cells express this genes at a level greater than 0, at least in terms of the RNA-seq data that we sampled and measured. P-val adjustment is just multiple comparisons correction. So again, for people who are perhaps less, that have less of a background in math and stats, when you have a statistical test and you test it, thousands or hundreds of genes, well, again, by the nature of the p-value, some of your results would be false positives. So we basically do an adjustment to essentially make all the p-values a bit bigger. And then we do our cutoff for what is significant. Ultimately, the point is to try to control and not have false positives. We can take a quick look at the top 5 genes. We can take a look at how these genes are expressed in our dot plot that we were looking at before, in our UMAP plot that we're looking at for clusters. So again, for those working with Binder, you did not run this code, so this part would not work. Hopefully you're able to follow along with what I show on screen here, or perhaps you can look at the published R Markdown document. But you can see in general, this is sorry, that was a different thing that we're going to see later. This is a heat map of all of our markers, our different subclasses. So you can see for oligodendrocytes, for example, these genes, which are hard to see because there's so many genes in this plot, but these genes that are rows are highly expressed and relatively specifically expressed in oligodendrocytes, compared to these genes that help us distinguish microglia versus these genes that are a part of OPC cells or oligodendrocyte precursor cells, and so on and so forth. So in general, we have some genes that are highly expressed and selectively and relatively specifically expressed in each cell subclass. These are the genes that we're going to use to estimate the different subclasses in the second tutorial that Keon is going to lead. We can run this code to basically get some kind of class label information as well. So for every subclass, we can see whether it's excitatory cells or inhibitory cells, for example. We can do a little bit of arrangement and write this as an output. So for those who are on Binder, this is, okay, so technically I did just output this, but even if I didn't, this human markers CSV file is already in the data folder. So hopefully you will be able to just read this in for the second tutorial and use that for the second tutorial. For this, and for this part, Binder folks should be able to follow along. This is what happens. Well, okay, what if we don't care about all of the subclasses? We only care about one subclass. We only want to take that one subclass and compare it to all the other subclasses. In this case, we would use a find markers function, and we would give it an identity. So we would, again, we would still use subclass as our main active identity, right? We're still looking at subclasses, But now, instead of every subclass, instead of looking for genes differentially expressed in every subclass, we only want to look at genes that are highly enriched in oligodendrocyte. So we can run that for our oligodendrocyte markers. When we do this run, again, we have a similar kind of output. Notice now that there's no longer a subclass column compared to before. Before we had subclass labels, but now we don't have subclass labels here because, again, this is a function that we specify one subclass. So these numbers, so these genes, we can definitely see in a few different ways. We can head it to see what our markers are, but violin plot would illustrate some of these as well. So we can see that in our oligodendrocytes, these genes are highly expressed, that's on the y-axis, whereas for these other cell subclasses, we either have little or no gene expression for these genes. We can also take a look at how these genes, in terms of a heat map, for how these genes look in that 2D UMAP plot that I mentioned previously. So for the ST18 gene, we can see it's highly expressed in this cluster. The PLP1 is highly expressed in this cluster. Again, there are some, it's not that they're only in those cells, but that they're highly and relatively specifically in these cells. And these cells, again, since we're looking at oligodendrocytes, we can go back up and take a look at our map. And what do you know? These are oligodendrocytes. So in our oligodendrocytes, through the functions that we just ran, we find these genes that are, surprise, surprise, highly and relatively specifically expressed in oligodendrocytes. Finally, just another variation on this. What if we don't care about, what if we want to look at only like 2 specific cell groups? I really just want to find genes that help me differentiate between SST cells and VIP cells. They are both inhibitory neurons. So, can I do that? And I'm talking about this, so of course you can. We can specify identity 2 for the comparator cell group in the same find all markers function. In the final markers function, everything else is the same as basically what we've used before, but now we can compare between SST and VIP. When we do this comparison, we are only comparing cells that have been classified as SST cells versus cells that are defined as VIP cells. We are no longer grouping all of the quote-unquote other cells together. We are no longer grouping, IT cells and PVALV cells. We're no longer grouping all of them into the other groups. We are only comparing gene expression between SST and VIP cells. So we can run those. And we can take a look at our results, formatted the same way. In this case, PCT1, of course, would be our SST cells, and our PCT2 would be our VIP cells. So 100% of SST cells express NXPH1 at a level greater than 0, but 42% of VIP cells express NXPH1 at a level greater than 0, at least in our data set. So this is the end of our SRAT tutorial. We will be taking the markers that we found for final markers, and we will be taking that into the second tutorial. This is where we will use these genes to try to help us estimate the cell type proportions of these different subclasses. What we have done here is taken single-cell RNA-seq data. We normalized our data. We did a simulation of finding clusters of cells, and we saw that they generally match very well with the subclasses defined by the Allen Institute. We took the subclasses from the Allen Institute, and we did statistical testing for every gene, or at least every gene that passed our thresholds, comparing between every cell group and all the other cells, resulting in us having a list of markers, a list of genes that are highly and relatively specifically expressed in each cell subclass relative to all the other cell subclasses. So this is the end of the tutorial, the first tutorial. I'm happy to take any questions, or we can take an early break, or I can help troubleshoot. Depends on what people are interested in. Thank you, Dad. Sridhar wants me to introduce myself. So I come from a web background. I did my postdoc. Sorry, I did my PhD in the Department of Pharmacology in the University of Toronto. I was working with essentially a mouse model of. We like to study it in the context of schizophrenia and perhaps sometimes autism spectrum disorders. Yeah, so I do not have a math background. I do not have a computer science background. I did have some education in coding, but yeah, in general, I think at a level where like at a level where you can be functional, at a level where it's serviceable. I do not think it is very hard for people to get started. Most of the time, people who are making these tools for people to use, they are oftentimes trying to make these tools approachable and usable and user-friendly. So it's very, it is approachable, it is fairly straightforward. It just takes some digging to find the resources. That is if you have a bit of background in coding. If you don't, starting with some basic coding is always a good starting point as well. I will say though, my progression from as a bioinformatician is basically during my PhD, my PI, Amy Ramsey, wanted to do some RNA-seq stuff. So we got some RNA-seq data, and then I was basically playing around in like a user interface web server browser. And basically all I did was like click around things and try to get things to work. Since joining Sridhar's lab, I have moved more towards like a coding context to access all of the tools that I use. And these are tools that I became like became gradually more aware of and gradually more familiar with, starting with my PhD. So starting with my PhD, we had our RNAseq data. I was using Use Galaxy. It's basically like a point-and-click interface online server that people can sign up for and use. But if they, instead of needing to write Linux code, everything is kind of point-and-click. So that was pretty nice. From there on, though, I was introduced to all of the different tools bioinformaticians use for aligning data and quantifying and doing statistical testing. So then from there on, I started reading into about all the different tools that people use. At that time, I was using the Cummerbund. But since then, just trying to understand those tools and just getting into the space, I learned more about Star and DSeq and Azure. And from then on, when we wanted to do more single cell stuff, when we wanted to do more clustering, we went to Seurat. So sometime in between that transition from using those point-and-click resources on Use Galaxy to using Star and Azure in R and Linux, there was a transition of going from that point-and-click into coding. So yes, and a lot of those fun names are puns. So a great place for people who love puns. So we only have about 8 minutes before this is officially done. I can take more questions, or maybe we can share the GatherTown link, and I'll join there after I get more water from my dying throat. Hopefully, I'll be able to hear people in GatherTown. Or if there's more questions, I can wait around here as well.
I just posted another poll asking about the pace of the tutorials and if it's too fast or too slow or just right. Yeah, sorry, Sonny, you're probably not able to hear me, but yeah, we have 4 votes for just right, so I hope we're doing okay. Yeah. More questions or comments?
I guess in that case, we can end it here.
I hope I'm not being asked a question because I still don't hear anything.
No. Okay, so why don't we end this broadcast? We're going to, just to sort of indicate the time, so it's in 20 minutes, we're going to start the second tutorial of the day. And Keon will, yeah, 2:45 Eastern time. So Keon's going to lead that tutorial. I think some of us will be in the GatherTown afterwards, and or in the intervening time. So we'll see you all at 2:45 Eastern for the next tutorial. Okay. Once again, let's thank Sonny. Okay. See you all very soon.